All right, so the purpose of this recording is just to kind of summarize the main points from Monday's lecture. What that doesn't mean is that only stuff in this video is going to be tested on and everything that's not in the video is not testable. Make sure that you go back through every slide when you are revising for the test. This is just a guide and at least in part a lecture to make up for the fact that I wasn't able to teach on Monday. So I'll be skipping over some of the slides and stopping at others. The first one I want to stop at is this slide here that contains the learning objectives. If you've never used these before, they are really helpful to study from. The learning objectives will be listed in every single lecture that I ever post online, and they basically contain a list of the most important points from every single lecture. So you can actually turn these into your own study guide if you like. Make sure that as you go through the lecture and as you're revising that material that you can answer all of these learning objectives, that you could write something out to explain or describe or compare or contrast wherever you have like different like verbs used in the learning objectives. They're a really great tool to see if you actually understand that content. So I would recommend stopping here. The next slide I want to skip to is the one that starts discussing the 3 stages of transcription. We ultimately have 3 steps in this process: initiation, elongation, and termination. Initiation is when the process begins. Elongation is the process that describes as RNA polymerase is moving along a transcription unit and making the RNA. And then termination is the term that's given to describe when transcription ends. So in this slide here, there's a few important things highlighted. One of them is the promoter region. The promoter is where RNA polymerase is going to bind. Remember that DNA is the starting material for transcription as well as it is for replication. So here's our DNA molecule. We've got 2 strands. So like in replication, our new nucleotide polymer of RNA is going to be made using a polymerase. But since we're making an RNA, it's actually an RNA polymerase, not a DNA polymerase. Also, like in replication, the new nucleotides can only be added in the 5' to 3' direction. One of the biggest differences between RNA polymerase and DNA polymerase is that DNA polymerase requires a primer to get started, but RNA polymerase doesn't. So it can begin making an RNA transcript, like it's doing down here, without the use of a primer. It can set that up on its own. Other important things that are labelled here, this green region, the promoter, you'll notice that within this region, this overlaps with the transcription unit. So the promoter region is where RNA polymerase will bind. There will be a transcription start site, and then transcription will continue until it reaches a transcription termination site or a transcription stop site. And this is what's known essentially as a transcription unit. So the promoter kind of helps to orient RNA polymerase and get it ready to begin transcription. Here's a closer version of what a promoter might look like in eukaryotes. You can see a sequence in here called the Tata box. And this is in eukaryotes, but not prokaryotes, critical for forming the initiation complex, so before transcription can actually begin. This sequence is recognized by RNA polymerase and it can bind and then proceed to initiate transcription downstream. What you've got going on here are transcription factors that are binding to the promoter region. It's not only RNA polymerase that binds here. Especially in eukaryotes, there are loads of extra proteins that actually help this process. Transcription factors usually bind first. And so they start to assemble on the DNA, and then as they assemble, then RNA polymerase can bind as well. So in bacteria, there's really only one RNA polymerase that transcribes all RNAs. In eukaryotes, there's actually multiple RNA polymerases, and this is something that we'll come back to later. But RNA polymerase 2, which is the one that's labelled in this diagram here, so here's RNA polymerase 2. This is the polymerase in eukaryotes that transcribes pre-mRNAs that will be used to make proteins. So now in this figure, we've gone from initiation of transcription, where our transcription factors are present, our RNA polymerase is present, they're bound to the promoter. Now we're in the elongation stage, where the RNA polymerase is now moving away from the promoter and it's starting to synthesize the new strand. So in this figure, transcription is moving from left to right. You can see the RNA polymerase is unwinding a little bubble in the double helix. About 10 to 20 bases at a time is what it unwinds. It constantly reads the next nucleotide in the template strand and then adds in a complementary RNA nucleotide. Here's what this looks like up close. In blue, we've got our DNA strands. And then in red, we've got our newly made RNA. It's being made 5' to 3'. So here's our 3' end up here. Here's the bubble, essentially, within this boundary here. We've got our transcription bubble. Now it's relating to transcription, so it's not a replication bubble, it's a transcription bubble. And then within this, we are reading the template strand of DNA, not we, RNA polymerase is, reading the template strand of DNA, and where it reads a T, it will add in an A. Where it reads an A, it will add in a U. Where it reads a G, it will add in a C, and so on. So this 3' end here is the growing end, where this A is going to bond to the T, this U is going to bond to an A. This is basically what elongation is. It's as the RNA polymerase moves along the template, reads the template strand, and then adds in complementary nucleotides. Once the RNA polymerase gets to the end of the transcription unit, it reaches what's called a terminator sequence. Termination in bacteria and eukaryotes work on different mechanisms. In bacteria is where you would see a terminator sequence. That's on the next slide. In eukaryotes, there's an alternative mechanism for ending transcription, where RNA polymerase 2 transcribes what's called a polyadenylation signal sequence, and then the RNA transcript is released about 10 to 35 nucleotides past that polyadenylation sequence. Here's what this termination structure looks like in bacteria. So during termination, the RNA polymerase receives a signal to stop transcribing. So here we're zooming in on a little segment of DNA that's being unwound by the RNA polymerase. And so here we've got in yellow our newly synthesized RNA strand. Here's its 5' end, and here it would be where its 3' end is. So what happens in bacteria is, as the sequence gets close, so the synthesized 5' to 3', as it gets close to the end of the molecule that's being made, there's a sequence that's built in that can fold back on itself and form this hairpin structure. The bases are colored according to this map over here. But what is in all of these bases here is guanines and cytosines. They're complementary. Even though they're on the same strand of RNA, this folds back on itself, and then base pairs form between these G's and C's. And remember that it's a strong base pair between a G and a C because it's 3 hydrogen bonds. So this hairpin structure is called a terminator structure. And then what follows is a stretch of U bases, or uracil bases. So this is a U. So the combination of this hairpin structure and then this stretch of U's causes the RNA polymerase and the DNA and the mRNA to become very unstable. And so they all break apart. and transcription stops. There's other mechanisms of termination in prokaryotes as well. And again, the mechanism in eukaryotes is different too, but since it's not very well understood, we're not going to spend too much time talking about that for now. On this slide, we can see an example at the top here of our chromosomal DNA. So imagine this is a DNA helix, this long molecule here. And then you can see a series of transcription units. So you've got promoter and the sequence and then the terminator. And then there's some regions between genes that are in purple. And then a promoter and then the coding sequence and then a terminator. And so on. So what's important to remember about all of this is that each gene has a specific directionality of transcription. And this is dictated by the promoter. So the DNA template strand will always be read 3' to 5', and synthesis of the RNA is always 5' to 3'. But the direction of transcription changes depending on the orientation of the promoter. The promoter has to come at the beginning of the gene. So notice how for gene A, here we've got promoter and then gene A, so transcription goes in this direction. Here we've got promoter and gene B, so the transcription goes again in the same direction. For gene C, the promoter is here, and then the gene sequence is here, and so transcription is going to go from right to left in the opposite direction. But in all cases, we read the template strand in the same way, and the mRNA is made 5' to 3'. Now I want to just come back to the 3 different polymerases that are present in eukaryotes in terms of RNA polymerases. And here's the kind of different promoters that they recognize that dictates what kinds of RNAs they transcribe. So in all of these cases, just remember, like the diagram over here suggests that I stole from a Nature review. In prokaryotes, it's a lot simpler than the situation in eukaryotes. Eukaryotes use so many more transcription factors. We'll look at gene regulation within this unit and you'll see how much more complex it is when it comes to eukaryotes. But for the 3 polymerases, Pol1 transcribes ribosomal RNA, Pol2 transcribes mRNA, this is the one that we talked about earlier, and most small RNA. Small RNAs are usually regulatory RNAs. And then Pol3 transcribes tRNAs, which we talked about in class today, and we will talk about them again on Friday in translation, and some small RNAs. So 3 different polymerases, 3 different sets of promoters that they recognize, and different types of RNAs that are produced. So we've done a few comparisons here between prokaryotes and eukaryotes. You might remember this slide from Unit 2, where we talked about prokaryotes and their basic cell structure, and we asked these 3 questions about where's replication happening, transcription, and translation. In prokaryotes, remember we talked about how they all happen in the same compartment because there's no organelles. So this is really important here because in prokaryotes, transcription is actually coupled to translation. This is honestly one of my favorite reasons that bacteria are just so efficient and amazing. Here, what you can see in this, the cartoon at the bottom and then the microscopy image at the top, these little strings here, like they are DNA, and it's blue in this figure down here. And then from this DNA, we've got RNA polymerase that is transcribing an mRNA, which is the kind of green squiggle. And then before that mRNA is even finished, you have ribosomes, which are these orange blobs, that bind to the mRNA. As soon as they find a start codon, they start translating. So this is called a polyribosome, where you've got, and that's what you can see in these figures here, these like beads on a string type structures. This is an mRNA that's being made from DNA. Before it's even finished, ribosomes are jumping on that mRNA, they're finding start codons, and they're beginning to translate. So here you can start to see all these kind of purple squiggles that are coming out. These are supposed to represent polypeptide chains that are emanating from this mRNA that's still being synthesized from the DNA. So these two processes, transcription and translation, happen literally at the same time. Sure, transcription happens a little bit before translation, but they can happen simultaneously in bacteria because all of this happens in the one compartment. In eukaryotes, though, DNA replication takes place in the nucleus, as does transcription. But translation happens in the cytosol. The cytosol is where we find all of our ribosomes. And so there has to be something different that happens here. The mRNA actually has to travel out of the nucleus and into the cytoplasm, where it can find the ribosomes, that's where the amino acids are located. They're just kind of floating around the cytoplasm, and all the machinery that will help make translation happen. So it makes sense then that there's some differences in eukaryotes in terms of how transcription and translation work. In eukaryotes, they have to prepare their mRNAs for export from the nucleus. So usually when eukaryotic mRNAs are made, they are made in a long structure that's known as a pre-mRNA that has to be processed to make a mature mRNA. What you can see here is a figure where you have a promoter, the terminator, and then you've got these color-coded sequences that are pink or dark pink and green. Exons, like this and this region here, these are the expressed regions. Introns, this guy and this guy, these are the intervening sequences. And so they actually have to be removed in a process called splicing. Introns are removed, the exons are spliced together, and then there's other modifications like a 5' cap is added and a poly A tail. And these types of things help stabilize the mRNA, and they help signal that it's ready for transport. So it happens in this order: capping and then tailing and then splicing. In terms of the cap, it's actually a modified guanosine. It's a modified nucleotide that's stuck on the 5' end. It's kind of stuck on almost upside down. It's a guanine nucleotide that's connected to the mRNA via an unusual 5' to 5' triphosphate linkage. You don't need to focus on that too much for now. Just know that the cap is really important for mRNA to be able to exit the nucleus, and it's also really important because this is where the ribosome can find an mRNA that needs translated and actually bind it. In terms of the tail is a poly A tail. So the poly A tail is a stretch. It can literally be from like 50 to 250 adenine nucleotides that are added. So this is actually not coded for by the gene, but each species will have a specific length of poly A tail. So this is our poly A tail here. Notice how it's not present after transcription, but then it's added by a special polymerase that's known as poly A polymerase, so not Pol2. This guy has a role in stability and protection from degradation by hydrolytic enzymes. So this helps to stabilize the mRNA for export from the nucleus. It also helps, it also helps the lifespan of the mRNA in the cytosol. It helps it live for a long enough time that it can be efficiently translated into a polypeptide. So if this is another schematic of our pre-mRNA and our mRNA, in the pre-mRNA we've got our 5' cap, we've got our poly A tail at the 3' end, and we've got exons that are in dark red here. There's 3 exons, and then our introns are in kind of light pink. the introns are cut out, the exons are spliced together, and so this guy down here would be our mature mRNA, or just quite simply an mRNA. But how do we actually get to this? How do we get to this structure where we remove these introns? This process happens using a complex of proteins and RNA that's called the spliceosome. So the spliceosome is assembled, is huge actually. It's assembled from small ribonucleoprotein particles that are abbreviated as SNRPs and literally pronounced like that, and about 80 proteins. So their job here is to recognize the boundaries between introns and exons, and then remove the introns and join the exons. So it creates an mRNA with a continuous coding sequence, and we get rid of all the intervening stuff. So the way that this works is in a kind of 4-step process. The first step, you have the SNRPs that recognize the intron-exon boundaries at the 5' end. So here's our 5' end, if you like. And then these blue blobs are going to represent our small ribonucleoprotein particles. So they recognize the intron-exon boundary at the 5' end and the branch site. So that's what's going on here. And then in the second step, they associate with other particles. So other particles start to bind and form the spliceosome. And they start to loop out this intron. So they're kind of bending the structure, if you like. Then we move towards starting to cut this guy out. The first place where the cut is made is on the 5' splice site. So the 5' end of the intron. So the 5' end of the intron is cut and it's removed, and then this loops from a position where it was joined down here, loops around, and it joins with this location called the branch site that's in part of the intron. And it forms this kind of half loop structure that's known as a lariat. And then all that remains is essentially to cut the 3' splice site, so to remove from this end essentially. So if you chop that completes the removal of the intron. It's kind of held in this like loop structure here by the spliceosome proteins, and then this gets degraded. What you'll also notice is that down here, in step 3, the 2 exons were brought close together, and then in step 4, these guys are now covalently bonded together. Happy days. What is really cool about this is that there is not one way only to make every single protein. There's a process that's called alternative splicing, and this creates different proteins literally from the same gene. So in this example here, this is one gene. Here's where transcription starts, and it continues all the way down here. There's 5 exons. And so here we're making our RNA through transcription, and then through alternative splicing, we must start with number 1, and we must end with number 5. But then we can splice 1 and 2 together, 2 and 3 together, 3 and 4 together, 4 and 5 together to make option number 1. We could splice 1 and 2 together, skip out 3 and then splice 2 and 4 together, and then 4 and 5 together to create this option here. Or we could do option C, I guess, where we have exon 1 splice to exon 2, skip out, exon 2 splice to exon 3, skip out number 4, oh gosh, and then splice exon 3 with exon 5, and so we end up with this structure here. So this can basically create 3 different proteins because they have 3 different combinations of these coding regions from one gene. Pretty neat.